ಶುರು ಮಾಡೋದ ಮೇಡಮ್?
Yes, sir. We'll begin.
ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರಗಳು. ಗುಡ್ ಈವ್ನಿಂಗ್ ಎವ್ರಿಬಡಿ. ಇದರ ವಿದ್ಯಾರ್ಥಿ ಪ್ರತಿಭಾಶೋಧ ಯೋಜನೆಯ 72ನೇ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಶ್ರೀಮತಿ ಲಕ್ಷ್ಮಿ ಮೇಡಮ್ ಅವರನ್ನು ಉಪನ್ಯಾಸ ಮಾಲಿಕೆಯ ವೇದಿಕೆಗೆ ಸ್ವಾಗತವನ್ನು ಕೋರುತ್ತಾ ಅವರನ್ನು ಈ ವೇದಿಕೆಗೆ ಮಾಡ್ಕೊಳ್ತಾ ಇದ್ದೇವೆ. ವೆಲ್ಕಮ್ ಟು ಯು ಮ್ಯಾಮ್.
Yes, sir. Thank you so much, sir.
ಅದೇ ರೀತಿ ಇಂದಿನ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಹಾಜರಿರ್ತಕ್ಕಂಥ ನನ್ನ ಎಲ್ಲ ವಿದ್ಯಾರ್ಥಿ ಮಿತ್ರರು, ಅಧ್ಯಾಪಕರು, ಶಿಕ್ಷಕರು ಹಾಗೂ ನನ್ನ ಸಹೋದ್ಯೋಗಿಗಳನ್ನು ಕೂಡ ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಸ್ವಾಗತಿಸುತ್ತಿದ್ದೇನೆ. ಹಾಗೂ ಲಕ್ಷ್ಮೀ ಮೂರ್ತಿ ಮೇಡಮ್ ಅವರ ಬಗ್ಗೆ ನಾನೇನು ಜಾಸ್ತಿ ಹೇಳೋದಿಲ್ಲ, ಯಾಕೆಂದ್ರೆ ಅವರ ಈಗಾಗಲೇ ಹಲವಾರು ಉಪನ್ಯಾಸಗಳನ್ನು ತಮಗೆಲ್ಲ ನಡೆಸ್ಕೊಟ್ಟಿದ್ದಾರೆ. ಜೊತೆಗೆ ಅವ್ರದೇ ಆದ ಒಂದು ಶೈಲಿಯಲ್ಲಿ ತಮಗೆಲ್ಲರಿಗೂ ಕೂಡ ಒಂದು ಕ್ವಶನ್ ಬ್ಯಾಂಕ್ ಪ್ರಶ್ನಾರ್ಥಕ ಪ್ರಶ್ನೆ ಬಂದ ಅಂತ ಹೇಳಿ ನಮ್ಮ JEE, CET ಮತ್ತು NEET ಎಕ್ಸಾಮ್ಗಳಿಗೆ ಅನುಕೂಲವಾಗುವಂತೆ ಭಾರಿ ಶ್ರಮ ವಹಿಸಿ ತಮ್ಗೆಲ್ಲರಿಗೂ ಕೂಡ ಒಂದು ಬುಕ್ನ ಪರಿಚಯ ಮಾಡಿಕೊಡ್ತಾ ಇದ್ದಾರೆ. ಅದನ್ನ ನಿಮ್ಗೆ ಡಿಸೆಂಬರ್ ಎಂಡ್ ಒಳಗೆ ನಾವು ತಮ್ಮ ಅಡ್ರೆಸ್ಗಳಿಗೆ ಕಳಿಸ್ತಾ ಇದೀವಿ. ತಾವೆಲ್ಲರೂ ಕೂಡ ಅದನ್ನ ಮರೆಯದೆ ತಮ್ಮ ಸರಿಯಾದ ವಿಳಾಸವನ್ನು ಕೊಟ್ಟು ಪಡ್ಕೋಬೇಕು ಅಂತ ಮತ್ತೊಮ್ಮೆ ತಮ್ಮೆಲ್ಲರಿಗೂ ಹೇಳುತ್ತಾ, ಮತ್ತೊಮ್ಮೆ ಸಲಾಮತಿ ಮೇಡಮ್ ಅವರನ್ನು ಸಾರಿ ಲಕ್ಷ್ಮೀ ಮೂರ್ತಿ ಮೇಡಮ್ ಅವರನ್ನ ಈ ಒಂದು ವೇದಿಕೆಗೆ ಸ್ವಾಗತಿಸುತ್ತಾ, ಓವರ್ ಟು ಯು ಮ್ಯಾಮ್.
Thank you so much, Siddhegowda sir. Yes, students, very warm good evening to all of you. And ivattu ನಾವು ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್ ಒಂದು ಟಾಪಿಕ್ ನ ನೋಡ್ತಾ ಇದ್ದೀವಿ. Particularly ಈಗ 11 ವರ್ಷದ್ದು ನೀವು, if you see the NEET papers from 2013 or you see the KCET papers ಇಲ್ಲ ಒಲಿಂಪಿಯಾಡ್ ಪೇಪರ್ಸ್ ಅಲ್ಲಿ ನಾನಿವತ್ತು ಮಾಡಬೇಕಾಗಿರೋ ಟಾಪಿಕ್ ಮೇಲೆ ಕ್ವೆಶ್ಚನ್ಸ್ 100% ಇಟ್ ಈಸ್ ಗೋಯಿಂಗ್ ಟು ಅಪಿಯರ್ ರೈಟ್. ಈಗ ಬರೋ NEET ಎಕ್ಸಾಮ್, KCET ಎಕ್ಸಾಮ್ ಆಫ್ 2024, there is a very good chance that this particular topic is going to be questioned. Apart from that, students, I'm sure you will note that for first year students, this is a very important topic for your board exam. Right, and I'm sure you will be able to do very well in this topic. Idu tumba interesting topic for a lot of students, so adre bagge ivattu maatadana. So what is the topic that we are going to do today? We will be looking at muscle structure, and I will be taking the topic forward in a little while, and I'm going to go to the structure of a myofibril. So muscle structure and myofibril is what we are going to be doing today. So delay madod beda. Where is this topic coming from? I'm sure our students know. Yaradru helboda? Tavu type ಮಾಡಿ ಕಳಿಸಿ, ಇಲ್ಲ unmute ಮಾಡಿಕೊಂಡು ಹೇಳಿ ನೋಡೋಣ. Which particular chapter of first year? ಈ chapter is important for first year students, and apart from that second year students ಗೆ ಬಹಳ ಮುಖ್ಯ because of competitive exams, correct? So any of the students will be able to tell me ಈ particular topic ಯಾವ chapter ಅಲ್ಲಿ ಬರುತ್ತೆ? Does it come in the animal tissues chapter, structural organization in animals ಅಲ್ಲಿ ಬರುತ್ತಾ, ಇಲ್ಲ ಇನ್ಯಾವ ಟಾಪಿಕ್ ಅಲ್ಲಿ ಬರುತ್ತೆ ಅಂತ ತಾವ್ ಯಾರಾದ್ರೂ ಹೇಳ್ಬೋದಾ? Animal tissues ಅಲ್ಲಿ ಸ್ವಲ್ಪ ಮಟ್ಟಕ್ಕೆ, correct Jairaj? Absolutely, you are right. So to some extent it does come under animal tissues, but muscle structure and myofibril structure has been explained in depth in the chapter called as locomotion and movement, right? Our particular chapter alli barutte. So let me not delay. I am going to start off with the first topic for today, which is muscle structure, right? Iga, when you take a particular, let us say I'm going to draw it diagrammatically. Let us say idu ಒಂದು bone ಅಂತ ಅನ್ಕೊಳ್ಳೋಣ. ನಾನು ಡಯಾಗ್ರಾಮ್ಯಾಟಿಕ್ ಆಗಿ ಡ್ರಾ ಮಾಡ್ತಾ ಇರೋದು ಸ್ಟೂಡೆಂಟ್ಸ್. ಸೊ ಲೆಟ್ ಅಸ್ ಕನ್ಸಿಡರ್ ದಿಸ್ ಇಸ್ ಯೋರ್ ಅಪ್ಪರ್ ಆರ್ಮ್ ಬೋನ್. ಅದರ ಹೆಸರು ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಹ್ಯೂಮರಸ್. ಓಕೆ, ಅಂಡ್ ಲೆಟ್ ಮೀ ಕನ್ಸಿಡರ್ ದಟ್ below this I'm going to have two bones of the lower arm anta ankolana. So I'm drawing two bones of the lower arm. So between these two bones, so basically it is as if the hand is being folded in this manner. ಈ ತರ hand fold ಆಗಿದೆ ಅಂತ I have shown it for you. So between these bones, you will find the presence of a big muscle. ಯಾರ್ಗಾರು ಗೊತ್ತಿದ್ಯಾ? Though ಅದು NCERT ಇಲ್ದೆ ಹೋದ್ರುನು, I'm sure our students are really good. Their general knowledge is good. E particular muscle, which is in contact with the upper arm bone and the lower arm bone. What is the name of this muscle? I'm sure many of our students will know. Nodi bega bega type ಮಾಡಿ ಕಳಿಸಿ. Let us see. So this muscle which is present between these two bones, there is a name for it. ಯಾರಿಗಾರು ಗೊತ್ತಿದ್ಯಾ ಏನದು ಹೆಸರು ಅಂತ? ನೋಡೋಣ ಯಾವ್ದಾರು ಮಕ್ಕಳು ಬರೀತಾರ ಟೈಪ್ ಮಾಡ್ತಾರ ಅಂತ. So this muscle is going to be connected to both the bones by means of a tissue. Let us see.
Lakshmita Shambhag ಹೇಳಿದ್ರು.
Yes sir.
Lakshmita Shambhag write on and send a message.
Sure. No problem, sir. No problem. Right. So this particular muscle is called as biceps. ನಾನು draw ಮಾಡಿರೋ muscle ಗೆ biceps ಅಂತೀವಿ. This muscle is called as biceps. ಇದ್ರ ಕೆಳಗಡೆ ಇರೋ muscle ಗೆ triceps ಅಂತೀವಿ. So this is the biceps muscle. So ಇದು biceps ಅಂತ ನಾನು ಕರಿಬಹುದು, or I can call it as a flexor muscle, right? ಇಲ್ಲಿ ನಾನು diagrammatic ಆಗಿ draw ಮಾಡ್ತಾ ಇರೋದು. So that is the biceps. We know that the muscle is connected to the respective bones by means of a tissue which is called as tendon. So illi ಒಂದು tendon ಇದೆ. At the top we have got a tissue which is called as the tendon. So that is the muscle which is joined to the respective bones. This particular topic for today, we are not going to be concerned about the bone structure. We are not concerned about tendon structure. Rather, we are interested in the structure of this tissue, which is the muscle, right? So let us not delay further at all, and let us go into the structure of a muscle. So biceps is the name of a muscle. So when we see the biceps, let us say we take the biceps like this. This is going to be the biceps, let us say, and this bicep is connected to the respective bone by means of the tissue which is called as tendon. And suppose I cut the muscle. When I cut the muscle, what is it that I'm going to see? Let us try and understand. So let us say the muscle is going to be cut. What is it that we are going to see in a muscle? So in a muscle, you find that the outermost layer of a muscle present in this manner. This outermost layer of a muscle which is seen is made up of fibrous connective tissue, which is very rich in the protein called as collagen. I'll repeat again. Every muscle, ಯಾವುದೇ muscle ಇರಲಿ, it may be the biceps, it may be triceps, ನಿಮ್ಮ ಬೆರಳಲ್ಲೂ ಚಿಕ್ಕ muscles ಇರುತ್ತೆ. So any muscle that you take, this muscle is going to be covered on the outside by a tissue which is fibrous connective tissue. And this fibrous connective tissue layer, which is rich in collagen, is called as muscle fascia. This is called muscle fascia. I'll repeat again. So the muscle is covered on the outside by fibrous connective tissue layer, which is rich in collagen fibers, and we call that layer as muscle fascia, because you need some kind of a tissue to attach the ligaments, to attach the tendons, to allow the nerves to come and innervate, correct? So which is that tissue? That is going to be the muscle fascia, which is rich in collagen. Okay, does anybody know an interesting fact about collagen? ಯಾರಿಗಾರು collagen ಬಗ್ಗೆ ಗೊತ್ತಾ? What type of biomolecule is collagen? Let us see if students are able to tell me. Collagen belongs to which kind of biomolecule, if you are able to tell me. And I want somebody else to type out as to what is an interesting fact. Guinness Book of World Records ಇದೆ collagen ಗೆ. ಯಾವ್ದದು? Collagen is famous with respect to what? It's a record holder for something. ನೋಡೋಣ, ಬೇಗ type ಮಾಡಿ ಕಳಿಸಿ, right? Yes, it's a protein. Correct, Lakshmita. It is a protein, no doubt about it. Collagen is a protein, and I'm sure Ms. Shanbag will also know something interesting about collagen. ಒಂದು record hold. Yes, Jairaj, it's correct. It's a protein. Okay, let's go ahead. ಅಷ್ಟರಲ್ಲಿ ಯಾರಾದ್ರೂ type ಮಾಡಿ ಕಳಿಸಿ. What is the record that collagen holds? Okay, adar bagge nodana, right? So this is the muscle fascia. What is this muscle fascia made up of inside? So when you see the muscle fascia, this is the cut muscle. I've taken the muscle and I've cut it. That is the muscle fascia. Inside this, I am going to find that there are a number of muscle cell bundles. Bundles of muscle cells are present inside this muscle in this manner. So there are a number of bundles that are present here inside. What I'm going to do is one of the bundles, I'm going to take it out from here. So I'm going to remove a bundle. Okay, it is like a pencil pack. And when the pencil na aache tegdange, I'm going to remove one particular bundle out like this. Okay, this is the bundle that I'm removing out. Everywhere else you find that there are these bundles that are present in this manner. And what are these bundles called as? These bundles that are present here are called as muscle fascicles. So these bundles are called muscle fascicles. Muscle fascia is the connective tissue covering of the muscle made up of collagen, rich in collagen. And the muscle will contain bundles of muscle cells, and these bundles of muscle cells are called as muscle fascicles. That is, so many muscle fascicles I have drawn. Iga obrobru, these bodybuilders, illa wrestlers, correct, or these professional sports people, ಅವರು ಸಿಕ್ಕಾಪಟ್ಟೆ ಜಿಮ್ಮಿಂಗ್ ಮಾಡ್ತಾರೆ. they do a lot of workouts. So ಅವ್ರದ್ದು ಬೈಸೆಪ್ಸ್ ನೀವು ನೋಡಿರ್ಬೋದು, ಬಾಹುಬಲಿ ಮೂವಿಲ್ ನೋಡಿದೀರಲ್ವಾ ಎಂತ ಬೈಸೆಪ್ಸ್ ಇದೆ ಅಂತ, ಕರೆಕ್ಟ್. ಸೊ ಆ ಒಬ್ಬೊಬ್ರೊಬ್ರು ಬೈಸೆಪ್ಸ್ ಎಷ್ಟು ದೊಡ್ಡದಾಗಿರುತ್ತೆ, ಒಬ್ಬೊಬ್ರು ಬೈಸೆಪ್ಸ್ ಚಿಕ್ಕದಾಗಿರುತ್ತೆ. ವಾಟ್ ಇಸ್ ದ ಡಿಫರೆನ್ಸ್? ದ ಡಿಫರೆನ್ಸ್ ಇಸ್ ದ ಮಸಲ್ ಇಸ್ ಒನ್. ಅವ್ರದ್ದು ಒಂದೇ ಬೈಸೆಪ್ಸ್ ಪರ್ ಆರ್ಮ್, ನಮ್ದು ಒಂದೇ ಬೈಸೆಪ್ಸ್ ಪರ್ ಆರ್ಮ್, ಬಟ್ ಅವ್ರದ್ದು ಹೆಂಗ ಅಷ್ಟೊಂದಿದೆ. ದೇರ್ ವಿಲ್ ಬಿ ಮೋರ್ ನಂಬರ್ ಆಫ್ ಮಸಲ್ ಫ್ಯಾಸಿಕಲ್ಸ್, and each muscle fascicle will have more number of muscle cells. Hangagi adu bulky. So we say they are bulking up the muscles, right? They are increasing the bulk of the muscles, right? So each bundle is called as a muscle fascicle. Muscle fascicle is made up of what? Muscle fascicle is basically going to be made up of muscle cells. So idar valagade yenide, inside this you find the presence of muscle cells, right? So idr ಒಳಗಡೆ ಎಲ್ಲ muscle cells ಇದೆ. ಒಂದು muscle cell na I'm going to take it out. One of the muscle cells I'll take it out. So this is filled with a number of muscle cells in this manner, and one of the muscle cells I have pulled it out, right? So this muscle cell is going to be packed in a bundle called muscle fascicle. Many muscle fascicles are packed together to form a muscle which is covered on the outside by a muscle fascia which is rich in collagen. Is that clear? E particular diagram ali, I have not drawn the nerves. There will be nerves innervating the muscle. Motor neurons ella barbekalla, right? Like that, there are many other types of neurons also which are going to penetrate the muscle. And apart from that, blood vessels will also be there, lymph vessels will also be there. So there will be other kinds of tissues also present in a muscle, but we are not interested in any of them. We are interested in this part of the muscular system, right? So now what we'll do is, I am going to take one of these muscle cells and try and magnify it in detail. And when I take a single muscle cell, what is it that I'm going to notice? A single muscle cell is going to be filled with a number of protein rods inside. A number of protein rods inside. When the protein rod na, I will take it out. That's a single protein rod which is going to fill up a muscle cell. And that protein rod is called as a myofibril. So this is a myofibril. This is going to be a muscle cell. The bundle of muscle cells is a muscle fascicle, and many muscle fascicles together make up the muscle which is covered by muscle fascia. So let us now go into details about this muscle cell and the myofibril. right? Adadmele, of course, I'm going to go into the details regarding myofibril. So idanna naniga alisbidtini because we don't need this now. Let me now go to the details about the muscle cell, muscle structure nodadvi, muscle cell, and then we are going to go into the myofibril. Now, when you take a muscle cell, I'm sure tissues chapter ಅಲ್ಲಿ Jairaj said that animal tissues chapter ಅಲ್ಲೂ muscle mention ಆಗಿದೆ, correct? It's not wrong at all. But ಈ chapter ಅಲ್ಲಿ ಇರುವಷ್ಟು detail ಆ chapter ಅಲ್ಲಿ ಕೊಟ್ಟಿಲ್ಲ. Just a brief introduction to muscle cell they have given. When you take any muscle cell, for example, you take a muscle cell, let us say this is a muscle cell. We know that skeletal muscle cells are going to be cylindrical and unbranched. They are cylindrical and unbranched in nature. So this is one muscle cell. So the outermost layer, it's an animal cell. An animal cell ali outermost boundary is going to be the plasma membrane. But this is part of the muscle cell, so it has got a special name. And this particular plasma membrane of a muscle cell is called as sarcolemma. The plasma membrane of the muscle cell is referred to as the sarcolemma. Lemma is the meaning of lemma means like a layer. Okay, so this is the outermost layer. Sarco andre ಏನು? ಎಷ್ಟು ಸತಿ you have heard about that word called sarco? ಯಾರಿಗಾರು ಗೊತ್ತಿದೆಯಾ? ಗೂಗಲ್ ಮಾಡದೆ ಹೇಳಿ. ನಿಮ್ಗೆ ಗೊತ್ತಿದ್ರೆ ಇಲ್ಲ ನಿಮ್ಮ ಟೀಚರ್ಸ್ ಹೇಳಿದ್ರೆ ಖಂಡಿತ you would have asked in the class. ಮ್ಯಾಮ್ ಸಾರ್ಕೋ ಅಂದ್ರೆ ಏನು? ಎಷ್ಟು ಸತಿ ಬರ್ತಾ ಇದೆ ಸಾರ್ಕೋ ಸಾರ್ಕೋ ಅಂತ? I'm sure you have asked your teachers. So ಅದು ಗೊತ್ತಿದ್ರೆ ಮಾಡಿ, ಬಟ್ ನಿಮ್ ಫೋನ್ ಇಂದ ಇಲ್ಲ ಲ್ಯಾಪ್ಟಾಪ್ ಇಂದ ಗೂಗಲ್ ಮಾಡಿ ನಂಗೆ ಹೇಳ್ಬೇಡಿ. ಅದು ಯಾರು ಬೇಕಾದ್ರೂ ಮಾಡ್ಬೋದು. ನೀವು ಹೇಳಿ ನೋಡೋಣ. Somebody tell me what is the meaning of sarco. Amele I had asked one more question. Collagen to record holder kelide. Biomolecules chapter ali, I'm sure students you have studied that collagen is the most abundant protein in the animal world. In the animal world, the most abundant protein is actually collagen. And both Lashmita as well as Jairaj have said that it is a protein, which is absolutely correct, right? So now this is sarcolemma and this is going to be the muscle cell. Many students, particularly NEET aspirants, those who want to write NEET or KCET, ಅವ್ರಿಗೆ confusion ಆಗುತ್ತೆ. Nerve fiber, nerve cell same. Muscle fiber, muscle cell same. Let me try and clarify that for you. Muscle cell is also called as a muscle fiber. So whether I use the word muscle fiber or I use the word muscle cell, both of them basically mean the same. There is no change there, right? Whereas nerve cell and nerve fiber is not the same. Nerve cell is the entire neuron. Nerve fiber is just the axon. So nerve fiber and nerve cell don't mean the same, whereas muscle cell and muscle fiber basically mean the same. So this is a muscle cell or a muscle fiber, and its outermost layer is called as sarcolemma, right? Now, the inside is going to be filled with cytoplasm, which we are going to call as sarcoplasm. This is called as sarcoplasm. Innu nange message bandilla. What is the meaning of sarco? Sarco anta prefix ನಾವು ಹಾಕೋದು ಯಾವುದಕ್ಕೆ ಅಂತ. So the word sarco is a prefix for flesh. ಈಗ ಯಾರಾದ್ರು ಒಂದು lion is eating the flesh of a deer andre, what is it basically eating? A lot of muscles it is eating. So the word sarco means flesh. Okay, so this is sarcoplasm, which is the cytoplasm present within the muscle cell. Now inside this, ಏನೇನಿರುತ್ತೆ? You will find that all the organelles that you need are going to be there. Like what? Nucleus irutta? Definitely. You will find that there will be nucleus present. These nuclei are present at the periphery. Illi nuclei are going to be present. Skeletal muscle cell is a multinucleate structure, or we call it as syncytium. The word that is used to say that it is multinucleate is syncytium. Okay, andre repeated karyokinesis ಆಗುತ್ತೆ, but cytokinesis ಆಗಲ್ಲ. So this kind of a structure is a multinucleate structure, which we call a syncytium, skeletal muscle cell. Plus ಇನ್ನೇನಿರುತ್ತೆ? ಒಳಗಡೆ you will find that here and there located, there is going to be special structures present here, which are called as sarcoplasmic reticulum. So ಇಲ್ಲಿ sarcoplasmic reticulum. ಏನಿದು ಹೊಸ ವರ್ಡು sarcoplasmic reticulum? Sarcoplasmic reticulum is going to be endoplasmic reticulum. Endoplasmic reticulum school ಇಂದಾನು ತಾವು ಓದ್ತಾ ಇದ್ದೀರಾ. So it is endoplasmic reticulum. Only thing is the difference between any other cells endoplasmic reticulum and the ER present here is this is going to be rich in calcium ions. So the difference is sarcoplasmic reticulum is ER present within the muscle cell, and the speciality is it is filled with calcium ions. So these sarcoplasmic reticula, you know, so many of them are also going to be present here. Okay, ಇನ್ನೇನಿರುತ್ತೆ? Of course, mitochondria have to be there, because mitochondria ಇಲ್ಲದೆ ಇದ್ರೆ ನೀವು how are you going to manufacture energy? Correct? So mitochondria definitely have to be there. So many mitochondria are going to be present, because muscle contraction and relaxation, both of them need ATP. Okay, students, please make a note. Okay, muscle relaxation is an easy process ಅಂತ ಅನ್ಕೋಬೇಡಿ, ಅದಕ್ಕೂನುವೆ ATP ಬೇಕಾಗುತ್ತೆ. So as a result, you will have a lot of mitochondria. Correct? Plus, ಯಾಕೆ ಇದನ್ನೆಲ್ಲ ಸೈಡ್ ಅಲ್ಲಿ ಡ್ರಾ ಮಾಡ್ಬಿಟ್ಟಿದೀನಿ? Why are the nuclei drawn at the side? Why is ER drawn at the side? That is a sarcoplasmic reticulum, mitochondria. Why are they all present in the side? That is because the inside of this particular muscle cell is going to be filled with a number of protein rods. Hodsati draw madidnalva just a few minutes ago. So there are a number of protein rods which are going to be present inside the muscle cell. I have pulled one out, and these protein rods are called as myofibrils. Okay, for example, ಒಂದು cylinder ತಗೊಳ್ಳಿ. Cylinder ತಗೊಂಡು fill it up with maybe some jelly, and ಅದರ ಮಧ್ಯದಲ್ಲಿ ತುಂಬಾ ನೀವು you put lot of sticks. When you put a lot of sticks, all the jelly comes to the side because you have filled it up with sticks. In the same way, when you pack it up with so many myofibrils, automatically what is going to happen? Automatically you find that the you know, cytoplasm, the organelles, all of them start moving to the periphery. Okay, that's why you find that these organelles are on the edges of the cell, right? So this is going to be what is a muscle cell structure. Right? Now we are going to go to this myofibril structure and try and see what is the myofibril actually made up of. Students would have noticed that I have kept on calling it as a protein rod. Makle, any of y'all can tell me ಯಾವ protein ಇದೆ ಒಂದು myofibril ಅಲ್ಲಿ ಅಂತ? Can anybody tell me? What are the types of proteins present in the myofibril? ನನಗೆ I do not know whether ನಿಮ್ಗಳಿಗೆ ನಿಮ್ಮ ಕಾಲೇಜ್ಗಳಲ್ಲಿ ಈ ಪರ್ಟಿಕ್ಯುಲರ್ ಚಾಪ್ಟರ್ ಆಗಿದ್ಯಾ ಇಲ್ವಾ ಅಂತ ನಂಗೆ ಐಡಿಯಾ ಇಲ್ಲ. But in any case, I'm asking, in case ಯಾರ್ದಾರು ಮುಗ್ದಿದೆ ಅಂದ್ರೆ you will be able to tell me. Okay, protein rod anta I have been calling the myofibril. Can somebody type out and send to me? ಯಾವುದು main proteins which are present in the myofibril ಅಂತ? Does anybody know? Let me check. ನಿಮ್ಗಳಿಗೆ ಗೊತ್ತಿಲ್ಲ ಅಂದ್ರೆ of course I'm going to go ahead. ನಾನು ಮುಂದೆ ಹೋಗ್ಬಿಟ್ಟು I'm going to start looking at the structure. So myofibril. Now I'm coming to myofibril. What are these? These are the protein rods which are present inside the muscle cell. So myofibril is made up of many types of proteins. ತುಂಬಾ ತರ proteins ಬೇಕು myofibril ಮಾಡಕ್ಕೆ. But among these proteins, most important proteins ಯಾವುದು ಅಂತ question ಬರುತ್ತೆ. So the most important proteins present in a myofibril are, we have actin and we have myosin. So the most important proteins present in a myofibril are of two types, that is actin as well as myosin. Apart from this, there are many other types of proteins also seen in a myofibril. Andmele, if I have to understand the structure of myofibril, it is very important that I first understand the structure of actin as well as myosin. So actin and myosin, if I'm able to actually, try and understand, then it becomes easy for me to understand the structure of a myofibril. So first actin and myosin ಬಗ್ಗೆ ತಿಳ್ಕೊಳ್ಳೋಣ, right? So if you are all ready, let me go to the structure of actin. ಆಮೇಲೆ myofibril automatically ತಿಳ್ಕೋಬಹುದು. So actin structure. Actin is going to be called, in biology, we call actin as a thin filament. Actin ge, we usually say it is the thin filament. What is actin made up of? ಈಗ ಒಂದು actin filament ಮಾಡಬೇಕಾದ್ರೆ what are the proteins that you need? Let us try and see. So if I have to create, okay, one actin filament, if I want to create one filament of actin, what are the various proteins that I need? Let us try and see. So the proteins that are needed to create this thin filament or actin filament are as follows. Let us see what they are one by one. So firstly, I will require the presence of spherical proteins in this manner. These spherical proteins are called as G-actins. What is this G-actin? G stands for globular actin. So first, in order to make actin, I require the presence of spherical proteins that are called as G-actin. G andre illi, it stands for globular actin. Okay. Next, what is required? Next, I will require the presence of a rod-like or thread-like protein, which is called as actin. Which is called as tropomyosin. I'm sorry. So you require a thread-like structure present here, which is called as tropomyosin. Students, don't get confused. Actin structure ಓದ್ತಾ ಇರುವಾಗ myosin ಅಂತ ಯಾಕೆ ಹೇಳ್ತಾ ಇದ್ದಾರೆ? I will come to that a little bit later. Okay. Next. So I need this kind of thread-like structure, which is called as tropomyosin. Adar jotege inyaudaru structure beka? Yes, I need another type of protein, which I'm going to draw like this, or we can draw like this. And this particular protein molecule is called as troponin. So I'll repeat again. So in order to make the actin filament, I will require spherical protein molecules called G-actin, thread-like protein molecules called tropomyosin, and these pyramidal or triangular 3D shaped protein molecules called troponin. ಎಷ್ಟು G-actins ಬೇಕು? ತುಂಬಾ G-actins ಬೇಕು. What I'm going to do to the G-actins? I'll take all the G-actins and I will attach them together. So when I attach them together, I'm going to get this ಒಂಥರ beaded chain-like structure I'm going to get. This beaded chain-like structure, which you can see, is called as actin, standing for filamentous actin. I'll repeat. So we take a number of G-actins and we are going to join them together. And when we join them together, we are going to get a long chain of beads. It will look like a long chain of beads, which is going to be called as F-actin. Ee thara, I'm going to take 2 F-actins. And I'm going to take 2 of tropomyosin. I'll take 2 F-actins, I'll take 2 tropomyosins, and what I'm going to do is I'm going to coil them around one another. Id ಎರಡು, that is F-actin ಎರಡು, tropomyosin ಎರಡು, I'm going to coil them around one another. So when I coil them, what is it that I'm going to get? This is the type of structure I'm going to get. So I will draw it on this side for your reference. I'm going to draw it on this side for your reference. This is how it is going to be. So I hope you got it. I've taken 2 of F-actin, I've taken 2 of tropomyosin, and I'm going to coil them together. This is how you're going to get. This is 1 F-actin, in this manner. This is another F actin. In this manner. So I have coiled them around one another. This is one tropomyosin, ilinda bandu. Hige, and ilinda bandu. Hige. So I have taken two F actins and two tropomyosins and I have coiled them around one another. When I coil them around one another in this manner, certain sites or pockets are created. The sites or pockets created by the coiling of these around one another is called as myosin binding sites. ಮತ್ತೆ ಹೇಳ್ತೀನಿ, when you take two of F actins, two of tropomyosin and coil them around one another, you are going to get some sites. Those sites are going to be called as myosin binding sites. Wherever you find there is a myosin binding site, for example, let us say ಇಲ್ಲಿ ಒಂದು myosin binding site ಇದೆ ಅಂದ್ರೆ ಅದರ ಮೇಲೆ ಒಂದು troponin ನ place ಮಾಡಬೇಕು, or it is present in nature in this manner. So wherever there is a myosin binding site, at that particular location there will be a molecule of troponin. ಇಲ್ಲಿ ಒಂದು site ಇದೆಯಾ, ಅದರ ಮೇಲೆ ಒಂದು molecule of troponin is present. ಇಲ್ಲಿ ಒಂದು ಸೈಟ್ ಇದ್ರೆ ಅದ್ರ ಮೇಲೆ there is a molecule of troponin present. ಇಲ್ಲಿ ಇದ್ರೆ ಇದ್ರ ಮೇಲೆ ಒಂದು. So wherever you find that there are these myosin binding sites, ಆ particular location ಅಲ್ಲಿ you have to place a troponin molecule or a troponin molecule is present. For example, let us say ಇದು myosin binding site ಅಂತ ಅನ್ಕೊಳ್ಳೋಣ. ಈ ತರ ಒಂದು ಸೈಟ್ ಇದೆ, ಅದ್ರ ಮೇಲೆ troponin molecule will be present. So itara ಎಲ್ಲೆಲ್ಲೆಲ್ಲಿ there is myosin binding site, troponin molecule will be present over it. So troponin molecule ಗೆ otherwise ಅದಕ್ಕೋಸ್ಕರ ನಾನು 2 copies ಬೇಕು, 3 copies ಬೇಕು ಏನು ಹೇಳಲಿಲ್ಲ, because it depends on how many such sites are present and what is the length of this coiled structure. Okay, so again I'll repeat, two F actins. F actins are themselves made of G actin. Two tropomyosins, coil them together. Wherever there is a myosin binding site, you mask it with a troponin molecule. Correct? Iga e class goskara, I am going to take this entire structure. What you see here, this whole thing that you see is going to be an actin filament. ಇದು actin filament. ಈ particular class ಗೋಸ್ಕರ I am going to represent this entire thing. ಈ class ಗೆ I am going to represent this whole thing as a yellow thin line. So the whole thing now onwards I am going to show as a yellow thin line. Andre idanna if we are able to magnify under an electron microscope, you will be able to see this kind of a structure. Right? You require high level of magnification in order to see it. So basically I'm representing this whole thing as one line. Now what I'm going to do is, E actin filament which is present, like this I'm going to take lots of actin filaments. Okay, this whole thing is one actin filament. Ee thara I'm going to take a number of actin filaments and I'm going to make them pass through a protein sheet. ಯಾವ ತರ, ಒಂದು ಶೀಟ್ ಆಫ್ ಪೇಪರ್ ತಗೊಂಡ್ಬಿಟ್ಟು ಅದರಲ್ಲಿ ನೀವು ಕಡ್ಡಿನ ಈ ಕಡೆಯಿಂದ ಪೋಕ್ ಮಾಡಿದ್ರೆ ಈ ಕಡೆ ಅರ್ಧ ಕಡ್ಡಿ, ಈ ಕಡೆ ಅರ್ಧ ಕಡ್ಡಿ, ಈ ಕಡೆ ಅರ್ಧ ಕಡ್ಡಿ, ಈ ಕಡೆ ಅರ್ಧ ಕಡ್ಡಿ ಬರುತ್ತಲ್ವಾ, ಆ ತರ ಯು ಟೇಕ್ ಅ ಪ್ರೋಟೀನ್ ಶೀಟ್ and you are going to pass all these actins through the protein sheet, or the protein sheet is going to help keep many actin filaments together. ಈಗ ಪ್ರೋಟೀನ್ ಶೀಟ್ ಹೇಗಿರುತ್ತಪ್ಪ, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ನಂದು ಬ್ಯಾಗ್ ಅಲ್ಲಿ there is a piece of paper. ಕರೆಕ್ಟ್, ಇದು ಪೀಸ್ ಆಫ್ ಪೇಪರ್. ಯು ಕ್ಯಾನ್ ಸಿ ದಿಸ್ ಪೀಸ್ ಆಫ್ ಪೇಪರ್. ಈ ಪೇಪರ್ ನಾನು ಹಿಂಗೆ ಸ್ಟ್ರೇಟ್ ಆಗಿ ಇಟ್ಕೊಂಡ್ರೆ full straight agi paper itkondre, you will be able to see only the line here. ನಿಮ್ಗೆ ಪೇಪರ್ ಏನು ಕಾಣಲ್ಲ, ಬರೀ ಒಂದು ಲೈನ್ ತರ ಕಾಣುತ್ತೆ, ಕರೆಕ್ಟ್ ಅಲ್ವಾ? ಅದೇ ತರ ಐ ವಾಂಟ್ ಯು ಟು ಇಮ್ಯಾಜಿನ್ ದಟ್ ದೇರ್ ಇಸ್ ಅ ಶೀಟ್ ಲೈಕ್ ದಿಸ್, ಬಟ್ ದ ಶೀಟ್ ಇಸ್ ನಾಟ್ ಪ್ಲೇನ್. ಈ ತರ ಪ್ಲೇನ್ ಶೀಟ್ ಅಲ್ಲ. ದಟ್ ಶೀಟ್ ವಿಲ್ ಬಿ ಲೈಕ್, ನೀವು ಲೇಸ್ ವೇಫರ್ಸ್ ಚಿಪ್ಸ್ ನೋಡಿದಿರಲ್ವಾ, ಲೇಸ್ ಚಿಪ್ಸ್ ಹೀಗೆ 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 ಇರುತ್ತಲ್ವಾ, ಲೈಕ್ ದಟ್ ಐ ವಾಂಟ್ ಯು ಟು ಇಮ್ಯಾಜಿನ್ ಅ ಪ್ರೋಟೀನ್ ಶೀಟ್. ಆ ಪ್ರೋಟೀನ್ ಶೀಟ್ ಅಲ್ಲಿ ದೀಸ್ ಆಕ್ಟಿನ್ ಫಿಲಮೆಂಟ್ಸ್ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ಅಟ್ಯಾಚ್ಡ್. So that what happens is, this is the structure you will get. So you will get, this is one actin, this is one actin, this is one actin, this is one actin, let us say. E actins are going to be joined by a protein sheet, and that protein sheet will appear like a zigzag line because that is the shape or that is the form of that protein sheet. So I have taken many actins and I have inserted them or joined them to this protein sheet. This particular protein sheet through which the actins are going to be joined together, this is called as the Z line. ಅದು Zwitterscheib line anta. It's a German word which kind of loosely translates into a zigzag line. It's called Zwitterscheib line. Okay, and it is the zigzag line that is going to be present there. It is a Z line, right, which is what you see. And these are individual actin filaments. These are individual actin filaments. Andre illi draw madidinalva, like that each one of them is. ಈಗ ಇದು ನಾನು ಮಾಡಿದ್ದನ್ನ ಈಗ ಸದ್ಯಕ್ಕೆ ಮರ್ತು ಬಿಡಿ. ಇವಾಗ ಏನಾಗಿದೆಯೋ ಅದನ್ನ ಈಗ ಲೆಟ್ ಅಸ್ ನಾಟ್ ಬಾದರ್ ಅಬೌಟ್ ಇಟ್, ಬಿಕಾಸ್ ಐ ಹ್ಯಾವ್ ಟು ನೌ ಕಮ್ ಟು ಮೈ ಮಯೋಸಿನ್ ಫಿಲಮೆಂಟ್. ಮಯೋಸಿನ್ ಫಿಲಮೆಂಟ್ ಬಗ್ಗೆ ಮಾತಾಡಿದ್ಮೇಲೆ ಅಗೇನ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಕಮ್ ಬ್ಯಾಕ್ to the actin filament, and let us see how actin and filament are going to be kind of put together in order to form myofibril. Adana innond ಸ್ವಲ್ಪ ಹೊತ್ತರಲ್ಲಿ ಬರೋಣ. But first let us go to our myosin filament and see how to make this myosin filament. Let us just try and understand. So this is how the structure is going to be. So I am now going to myosin filament. I'm going to go to myosin filament. So myosin filament, it is called as thick filament. Myosin ge thick filament anta we call, because it is going to be thicker under the electron microscope in comparison with actin filament, and that's why we call it as thick filament. Okay, ಒಂದು myosin filament ಮಾಡಬೇಕಾದ್ರೆ what do we need? In order to make a myosin filament, I require certain monomers. Monomers andre they are like building blocks. ಒಂದು wallದು monomer is like a brick ಅಂತ ಅನ್ಕೊಳ್ಳಿ, ಆ ತರ. Like that, in order to make myosin filament, which is a thick filament, I require some units. Those units are the monomers, correct? What are the monomers that are present here? So the monomers that are present to make myosin are called as meromyosin. They are meromyosin. Biologically, MER ಅನ್ನೋದು ಯಾವ್ದಾದ್ರು word ನಿಮ್ಗೆ either as a prefix or as a suffix ಕಾಣಿಸಿದ್ರೆ it will mean part ಅಂತ. Okay, so this is the small part of the myosin. It is like a component of the myosin. So we require meromyosin. Meromyosin are going to be of two types. What are the two types of meromyosin? One meromyosin has got low density. It is called light meromyosin, LMM. Correct? Hege kanutte idu? This is how it is going to look like. This is the light meromyosin which is present like this. Idu light meromyosin. Ee thara irutte. Okay, next type of meromyosin yaudu? It is called HMM, heavy meromyosin. That means its density is going to be more. Ade hege kanutte? Ee tara kanutte. Hige kanutte. So light meromyosin will look like this. Heavy meromyosin will look like this. Ee heavy meromyosin na we are going to attach to the light meromyosin. When we attach them together, this is how the structure is going to be. So the structure is light meromyosin, heavy meromyosin put together. That is going to be one of the bricks or components of myosin filament. When I see this structure, this is called as the tail. This is called as the cross link, or it is called as the neck. Different biologists give it different types of names, right? And then on the top, I'm going to have a head. So I have like a tail, and then there is a slight neck-like structure, and then there is going to be the head. When I see this head, suppose I see this head in this manner. When I see the head here, what is it that I'm going to notice? This head has got two different sides. Sides andre they'll be like it's a keyhole ideyala. Keyhole is like a sight in the lock. Athara, there is going to be some sights that are present. What are these two sights? So the sights that are present here are as follows. One site is called as ATP binding site. One site is called as ATP binding site. Basically, it is ATP breaking enzyme or ATPase. So that head, one of the components is going to be an ATP breaking enzyme, which is called ATPase. ATPase ge ATP join agbekalva. That's why it is also called as ATP binding site. The other site which is present is called as the actin binding site. Actin ಮೇಲೆ ಏನಿತ್ತು myosin binding site ಇತ್ತು. Myosin ಮೇಲೆ ಏನಿರುತ್ತೆ actin binding site is going to be present. So these are the two sites present on the head. Okay, this was a meromyosin. How to take many meromyosins and put them together? Let us try and understand. Okay, so idu ಒಂದು meromyosin. Now I have to take many meromyosins and I have to join them together. ಹೇಗಿರುತ್ತೆ ನೋಡಿ, this is one meromyosin. Okay, ಈ meromyosin ಗೆ ಇನ್ನೂ ಒಂದು meromyosin starts getting coiled in this manner. ಇನ್ನೊಂದನ್ನ coil ಮಾಡೋಣ. ಇನ್ನೊಂದನ್ನ coil ಮಾಡೋಣ. ಇನ್ನೊಂದನ್ನ coil ಮಾಡೋಣ. ಈ ಕಡೆ ಈ direction ಅಲ್ಲಿ coil ಮಾಡೋಣ. Like this. So I will coil them up in this manner. I'll take all of them and I'll start coiling them up like this. So some are coiled, our tails na coil madidre, some are coiled with the heads facing that side, 360 degree. Some are coiled with the heads facing the side, 360 degree. This is going to be a myosin filament. So this whole thing is one myosin filament. ಈಗ ಈ ತರ ಡ್ರಾ ಅಂತೂ ಮಾಡಕ್ಕಾಗಲ್ಲ. How do we draw it in a diagrammatic manner? So diagrammatically, from now onwards, for this class, I am going to show this structure. Hege actin I showed it as a yellow line. Ade thara, I am going to show this structure as a line. And heads na, I am going to show them like this. Okay, so for this class, I'm going to show this in this manner, which is a thick filament. Iga ee thara, I'm going to take lots of thick filaments and have to attach them to a sheet. Just like we took actins and attached them to a zigzag sheet, in the same way, I'm going to take these myosins and attach them to a sheet. But that is a straight sheet. It's a straight protein sheet. How are the attachment? This is one myosin. This is another myosin. Let us say in this manner. Okay, ಎರಡನ್ನ draw ಮಾಡ್ತೀನಿ for the sake of this explanation, and I'm going to attach them in this way. I will just attach them to a straight protein sheet. This protein sheet which is present, it is called as the Mittelscheiblein. Again, it's a German word. Basically, it is like the midline. Okay, and we just have to remember it as the line. ನಿಮಗೆ ನೀಟಲ್ಲಿ ಆಗ್ಲಿ KCET ಆಗಿ nobody will ask you for the Zwitterscheiblein, Mittelscheiblein, but you are expected to know Z line and M line, right? This is called as the M line. Is that clear till here? ಎಲ್ಲ ಅರ್ಥ ಆಗಿದೆ ಅಂತ ಅನ್ಕೊಂಡಿದ್ದೀನಿ. Now what I have to do is, ಈ ತರ I will take few parts, that is myosins attached to the M line or the protein sheet, actins attached to the Z line, that is going to be the the zigzag protein sheet, and I'll assemble them together. When I assemble them together, you will get myofibril, which we can then pack into the muscle cell. ಅರ್ಥ ಆಯ್ತಾ? So I'm going to go ahead. ಎಷ್ಟು ಆಗ್ಲೇ 4:45 ಆಗ್ತಿದೆ. Very simple. I'll just put it together so that ನಿಮ್ಮ myofibril ready ಆಗುತ್ತೆ. Okay? Right? So please remember, you have lots of different proteins present here, but what's most important is that the most important proteins are going to be your actin and myosin, because they are the contractile proteins. Baki proteins I need, I'm not saying we don't need them. Definitely the other proteins we are interested in, but the most important proteins for us, for muscle contraction's sake at least, is going to be your actin and myosin. So let us try and put them together. Aita? So ಈಗ I am now going to go to myofibril structure. ಒಂದು myofibril structure ಗೆ ಹೋಗ್ತಾ ಇದ್ದೀನಿ. Right? So let us see how this myofibril is going to be. See, very simple. Okay, I'll put it here. This is an actin. So I have many actins present here. Okay, and ಈ actins ನ ಹೇಗೆ join ಮಾಡಬೇಕು ನಾನು? I have to join them through a zigzag line in the middle. Madna, okay. Now what I'm going to do, I'm going to come a little away and draw one more such a group. ಈ ತರ ಒಂದು group. You can see many actins. It's a 3D structure, of course. ನಾನು 3D ಹೇಗೆ ಕಾಣುತ್ತೆ ಅಂತ ತೋರಿಸ್ತೀನಿ ಇನ್ನೊಂದು ಸ್ವಲ್ಪ ಹೊತ್ತರಲ್ಲಿ. And that is the zigzag line that is present, correct? Or the Z line, Zwitterscheib line. ಆಮೇಲೆ ಈ ಕಡೆ I'm going to go to the next actin. ಇದು next actin. Okay, and I am going to join them with a zigzag line. Right? Next, I'll go to one more set. ಇಲ್ಲಿ ಇನ್ನೊಂದು ಸೆಟ್ ಬಂದ್ಬಿಡ್ತೀನಿ. This one. And we are going to have all of these connected by the Z line. So I have drawn 3 sets of actins. ನಿಮ್ಗೆ ಕಾಣುತ್ತೆ ಇದು, which is thin filament connected through the Z line. Then I'm going to come through the myosin. Okay, myosin ನ ಹೇಗೆ assemble ಮಾಡಬೇಕು? If you all remember, myosins are going to be attached via the M line. Correct? Now see how I'm assembling them together. Myosin ನ ಹೀಗೆ assemble ಮಾಡ್ತೀನಿ. I will assemble it in such a way that part of the myosin edges are overlapped by the actins. So ಈ ತರ I'm going to assemble them together. Like this, I'm going to assemble them together. So that part of the myosin is overlapped by the actin. ಕಾಣಿಸ್ತಿದ್ಯಾ? This is the next one. Right? And connect them via a M line. Then this side, ಈ ಕಡೆ full draw ಮಾಡಲ್ಲ, I'll draw it partly. And that is going to be connected via the M line. Again, this side we'll go ahead. And this side. So I have drawn so many sets here. ಇದಕ್ಕೆ ಒಂದು M line ಹಾಕ್ಬಿಡೋಣ. ಇದಕ್ಕೆ ಒಂದು ಎಂಲೈ ಹಾಕ್ಬಿಡೋಣ. Right? So I have put all these now together. When I arrange them, I get a myofibril. ಇದು 2D structure. 3D structure ಹೇಗಿರುತ್ತೆ ಅಂತ ನೋಡ್ಬೇಕಾ ತಾವು? Though you don't need it for board exam or NEET, but it's very interesting. ನಿಮ್ಮಲ್ಲಿ ತುಂಬಾ ಜನ ಮಕ್ಕಳು ಮುಂದೆ, you may be working probably in the field of sports. You need to know about how muscles work. Maybe you are some team sports specialist ಆಗ್ಬೋದು, ನೀವು ಮುಂದೆ ಡಾಕ್ಟರ್ಸ್ ಆಗ್ಬೋದು, ರಿಹ್ಯಾಬಿಲಿಟೇಷನ್ ಡಾಕ್ಟರ್ಸ್ ಆಗ್ಬೋದು, ಸೋ ಮೆನಿ ಫಿಸಿಯೋಥೆರಪಿಸ್ಟ್ ಎಲ್ಲ ಆಗ್ಬೋದು. ಸೊ ಯಾವ ತರ ಇರುತ್ತೆ ಗೊತ್ತಾ? ಇದನ್ನ ನಾನು ತಿರುಗಿಸ್ಬಿಟ್ಟು ನೋಡಿದ್ರೆ ಹೇಗಿರುತ್ತೆ? This is ಮಯೋಸಿನ್. ಇದು ಮಯೋಸಿನ್. ಸೈಡ್ ಇಂದ ನೋಡ್ತಾ ಇದ್ದೀನಿ, ನಿಮ್ಗಳಿಗೆ ಗೊತ್ತಾಗ್ಬೇಕು ಮಕ್ಳು ನಾವೇನ್ ಹೇಳ್ತಾ ಇದ್ದೀನಿ ಅಂತ. ದಿಸ್ ಥಿಂಗ್ ಐ ಆಮ್ ಸೀಯಿಂಗ್ ಇಟ್ ಫ್ರಮ್ ದ ಸೈಡ್. ಅಂದ್ರೆ ಈ ತರ, ಇದು ಈ ತರ ಇದೆ. ನಾನು ಅದನ್ನ ಹೀಗೆ ತಿರುಗಿಸಿ ನಿಮ್ಗೆ ತೋರಿಸ್ತಾ ಇದ್ದೀನಿ. ಅರ್ಥ ಆಯ್ತಾ? ದಿಸ್ ಇಸ್ ಮಯೋಸಿನ್. ಮಯೋಸಿನ್ಗೆ ಏನಿದೆ? ಈ ಕಡೆ ಎಲ್ಲ ಹೆಡ್ಸ್ ಇದೆ, ಕರೆಕ್ಟಾ? ಹೆಡ್ಸ್ ಆರ್ ಆನ್ ಆಲ್ ಸೈಡ್ಸ್. ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿ, ಕರೆಕ್ಟಾ? ಇದೇನ್ ಗೊತ್ತಾ? ಆಕ್ಟಿನ್. ಇದು ಆಕ್ಟಿನ್. ನಿಮ್ಗೆ ಆಕ್ಟಿನ್ ಇಲ್ಲಿ ಮಾತ್ರ ಕಾಣಿಸ್ತಾ ಇದೆ. ಬಟ್ ಆಕ್ಟಿನ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ಹಿಯರ್, 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 ಹಿಯರ್. All around actins are present. ಅದು ಈ ಡಯಾಗ್ರಾಮ್ ಅಲ್ಲಿ ಕಾಣಲ್ಲ. This is how it is going to be arranged. ಹಂಗೆ ನಿಮ್ಗೆ 3D protein rod ಕ್ರಿಯೇಟ್ ಆಗುತ್ತೆ. ಅರ್ಥ ಆಯ್ತಾ? Now let's not worry about this. ಈಗ ಇದಕ್ಕೆ ಲೇಬಲ್ ಮಾಡೋಣ, right? So that we can end the class. ಸರಿ, ಏನು labeling? Let us try and see. Now when you see this under electron microscope, okay, you will find that the way they take up light, reflect light, etc., you will have slight variations because there is the thicknesses of the filaments are different, the way they are arranged are different, etc. Hangagi they are going to take in light and reflect light in different ways. Adella nodadmele scientists have been able to give certain names to the parts that we see because of the assembly of the actins and the myosin together. Inna nodtare, I want you to concentrate, dear students, on this part. ಈ ಪಾರ್ಟ್ ಮೇಲೆ ಕಾನ್ಸಂಟ್ರೇಟ್ ಮಾಡಿ. ಈ ಪಾರ್ಟ್ ಅಲ್ಲಿ ಅಂದ್ರೆ ಬರೀ ಇಲ್ಲಿರೋ ಪಾರ್ಟ್, ಬರೀ ಈ ಪಾರ್ಟ್ ನೋಡಿದ್ರೆ ತಮ್ಗೆ ಏನ್ ಕಾಣುತ್ತೆ? ಓನ್ಲಿ ಆಕ್ಟಿನ್ ಯು ಆರ್ ಏಬಲ್ ಟು ಸಿ. ಈ ಪಾರ್ಟ್ ಅಲ್ಲಿ ಮಯೋಸಿನ್ ಇಲ್ಲ. This is going to be the light band, or it is called as the isotropic band or I band. It is the light colored band and it is called as the isotropic band or the I band. Okay? Amele, I want you to now concentrate on this part. ಈ ಪಾರ್ಟ್ ಮೇಲೆ ಕಾನ್ಸಂಟ್ರೇಟ್ ಮಾಡಿ. ಈ ಪಾರ್ಟ್ ಅಲ್ಲಿ ಅಂದ್ರೆ ಇಷ್ಟು ನಂದು ಕೈ ಒಳಗಡೆ ಇರೋದು ಇಷ್ಟು ಪಾರ್ಟ್ ಏನ್ ಕಾಣಿಸುತ್ತೆ? ಯು ವಿಲ್ ಫೈಂಡ್ ಮಯೋಸಿನ್ ಇಸ್ ದೇರ್, ಬಟ್ ಆಕ್ಟಿನ್ ಇಸ್ ಆಲ್ಸೋ ದೇರ್. ಬಿಕಾಸ್ ಇಲ್ಲಿ ಓವರ್ಲ್ಯಾಪ್ ಆಗಿದೆ ಅಲ್ವಾ? ಫುಲ್ ಇಲ್ದೇ ಇರ್ಬೋದು, ಬಟ್ ಆಕ್ಟಿನ್ ಇದೆ ತಾನೆ. ಓಕೆ, ಸೊ ಯು ಹ್ಯಾವ್ ಆಕ್ಟಿನ್ as well as myosin present, no? So this is going to be of two types of proteins present here. So the way it takes in light, reflects light, etc. is going to be little different. This is called as an isotropic band or A band. Okay, bari A anta label martini. An isotropic band or A band which has got both myosin as well as actin. Iga ee A band alli I want you to concentrate on this region. Andre ee madhyad part matra concentrate ಮಾಡಿ, where myosin is not overlapped by actin. ಆ ಪಾರ್ಟ್ ನ ನೀವು if you concentrate here, you will find that this particular region which is present has got only myosin. ಬರೀ myosin ಇದೆ, ಕರೆಕ್ಟಾ? That part is called as the H zone. Some scientists call it as hyaline zone, or it actually stands for Henson's zone, which is the middle part made up of only myosin. Actin is not there. A band the madhyad regionally you will find this H zone present where only myosin is seen. That is going to be the middle part of the A band. idr madhyadalli I am able to find the M line. And I band du madhyadalli I am able to see the Z line or the Z line. Okay, so what you are able to see is going to be like this. Idanna draw madidre heegirutte nodi. You will find light band, and then there will be dark band, then there is light band, then there is dark band, light band, dark band. ಈ ತರ ಹೋಗ್ತಾ ಹೋಗುತ್ತೆ. ಈ ತರ ಹೋಗ್ತಾ ಹೋಗುತ್ತೆ. Light dark, light dark. Middle of the dark there is a small little area which is dark but slightly less dark than the rest. ಈ ತರ. Like this. It is dark but a little less dark than the rest. ಈ ತರ ಇದೆ. You understand? Can you see this? So this is going to be I. This is going to be A. And middle of A is going to be H zone. Illi en baratte? Zigzag line. Zigzag line. Illi en baratte? M line. M line. That is how the structure is going to be seen. So you have alternating light band, dark band, light band, dark band, which gives that striations to skeletal muscles, striped muscles annodu because of this alternating I band and A band, right? Ivaga, if we see this structure, ಈ ಸ್ಟ್ರಕ್ಚರ್ ಅಲ್ಲಿ ದೋ ಇಟ್ ಇಸ್ ನಾಟ್ ಪಾರ್ಟ್ ಆಫ್ ದ ಟಾಪಿಕ್, ರೈಟ್? ಮುಂದೆ ಯಾವಾಗ್ಲಾರು ಟೈಮ್ ಸಿಕ್ಕರೆ ನೋಡೋಣ, ಮಾಡ್ಬೋದು. ಇಟ್ ಇಸ್ ನಾಟ್ ಟುಡೇಸ್ ಟಾಪಿಕ್. ಹಾಫ್ ಆಫ್ ಎನ್ ಐ ಬ್ಯಾಂಡ್ ನೋಡಿ ಇಲ್ಲಿ, ದಿಸ್ ಇಸ್ ಹಾಫ್ ಆಫ್ ಎನ್ ಐ ಬ್ಯಾಂಡ್. ಕಾಣಿಸ್ತಾ ಇದೆಯಾ? ಒನ್ ಫುಲ್ ಎ ಬ್ಯಾಂಡ್ ಅಂಡ್ ಹಾಫ್ ಆಫ್ ದ ನೆಕ್ಸ್ಟ್ ಐ ಬ್ಯಾಂಡ್. ಇದು ಹಾಫ್ ಆಫ್ ಐ ಬ್ಯಾಂಡ್. This is one full A band. This is half of the next I band. So Z line to Z line, which includes half of I, 1A, half of I, is called as the sarcomer, which is the unit of contraction. Sarcomer is the unit of contraction. So muscle contraction theory, which is the sliding filament theory, is able to say that if the two Z lines come closer, sarcomer has contracted, which means the muscle has contracted. But that is, of course, another different topic. Okay, so that is going to be the structure of a myofibril, right? Yes, Sidegowda sir, in case the students have, ಏನಾದ್ರೂ ಡೌಟ್ಸ್ ಅದು ಇದು ಇದ್ರೆ ಕೇಳಬಹುದು. Yes, students, if you have any doubts, you can ask me any questions that you want. Maybe ನಾನೇ ನಿಮ್ಗೆ ಒಂದು ಕ್ವೆಶನ್ ನ ಕೊಡಬಹುದು. You will be able to, let's see if you are able to answer some questions. So ಒಂದು ನೀಟ್ ಕ್ವೆಶನ್ ನ ಕೊಡ್ತೀನಿ, ಒಂದು ನೀಟ್ ಕ್ವೆಶನ್ ನ ಕೊಡ್ತೀನಿ. Enzymatic activity that produces inorganic phosphate and ADP is present in which part of the myofibril? Enzymatic activity to produce by hydrolysis inorganic phosphate and ADP is going to be present in which part of the myofibril? So the options 1, 2, 3, 4 are option 1, troponin, option 2, tropomyosin, option 3, HMM, and option 4 is going to be F-actin. ಮತ್ತೆ ಹೇಳ್ತೀನಿ, enzymatic action to hydrolyze and produce inorganic phosphate and ADP is going to be present in which part of the myofibril? And the options are option 1, troponin, option 2, tropomyosin, option 3, HMM, and option 4 is F-actin. Anybody wants to answer this question? Type ಮಾಡಿ ಕಳಿಸಿ. I will go to the chat so that ನೀವು ಆನ್ಸರ್ ಮಾಡಿದ್ದನ್ನ ನೋಡಬಹುದು ನಾನು. Anybody? ನೋಡೋಣ. ಜಯರಾಜ್ ಫಸ್ಟ್ ಟೈಪ್ ಮಾಡ್ತಾನಾ ಇಲ್ಲ ಲಕ್ಷ್ಮಿತಾ ಮಾಡ್ತಾಳಾ? ನೋಡೋಣ. Let us see. You got the question? Basically, inorganic phosphate and ADP produce ಆಗ್ಬೇಕಾದ್ರೆ ATP has to break down. ATP break down ಆಗ್ಬೇಕಾದ್ರೆ ATP has to bind. So ಅದರಲ್ಲೇ ನಿಮ್ಗೆ ಆನ್ಸರ್ ಬಂದ್ಬಿಡುತ್ತೆ ಎಲ್ಲಿದೆ ಅಂತ. Let us see if you are able to answer. ಈಗ ಆಗ್ಲಿಲ್ಲ ಅಂದ್ರೆ you can send the answer to Sidegowda sir later. Okay? Right, sir? Over to you, sir.
Thank you, madam.
Thank you, sir. Thank you.
Chat box ಅಲ್ಲಿ ಯಾವುದೇ ತರ questions answers ಇಲ್ಲ madam.
Yes, that's fine, sir. That's fine. That's fine. No problem.
I sent through mail, madam.
Yes, sir. Yes, sir. Sure, sure, sure.
So I would like to thank you immensely for delivering very informative and insightful talk on muscle and myofibril structure.
Thank you, sir. Thank you.
On behalf of KST and myself, thank you, madam.
Thank you so much, sir. Thank you.
ಸರಳವಾಗಿ ಮಕ್ಕಳಿಗೆ ಕನ್ನಡ ಮತ್ತು ಇಂಗ್ಲಿಷ್ ಎರಡರಲ್ಲೂ ಕೂಡ ಹಲವಾರು ಉದಾಹರಣೆಗಳ ಮುಖಾಂತರ ಭಾರಿ ಸರಳವಾಗಿ ತಿಳಿಸ್ಕೊಟ್ಟಿದ್ದೀರಾ. ಮತ್ತೊಮ್ಮೆ ತಮಗೆ once again thank you, ma'am.
Thank you so much, sir. And ಯಾವ್ದಾದ್ರು ಮಕ್ಕಳಿಗೆ ಏನಾದ್ರು questions ಇದ್ರೆ ನೀವು ನಮ್ಮನ್ನ ಕಳಿಸ್ಕೊಡಿ.
ಖಂಡಿತ ಕಳಿಸ್ಕೊಡ್ತೇನೆ.
Thank you, sir. Thank you. All the best, dear students. Thank you.
ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಹಾಜರಾಗಿದ್ದಾರೆ ಎಲ್ಲ ವಿದ್ಯಾರ್ಥಿ ಮಿತ್ರರು, ಅಧ್ಯಾಪಕರು, ಶಿಕ್ಷಕರು ಹಾಗೂ ನನ್ನ ಸಹೋದ್ಯೋಗಿಗಳನ್ನು ಕೂಡ ಧನ್ಯವಾದಗಳನ್ನು ತಿಳಿಸುತ್ತಾ ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮವನ್ನು ಮುಗಿಸುತ್ತಿದ್ದೇವೆ. ಹಾಗೂ ನಾಳಿನ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಡಾಕ್ಟರ್ CK ಮಂಜುನಾಥ್ ಸರ್ ಅವರು ಕೆಮಿಕಲ್ ಬಾಂಡಿಂಗ್ ಎಂಬ ವಿಷಯದ ಮೇಲೆ ಉಪನ್ಯಾಸ ನೀಡಲಿದ್ದು ಅಂದು ಸಂಜೆ ಎಂದಿನಂತೆ 4 ಗಂಟೆಗೆ ತಾವೆಲ್ಲರೂ ಹೆಚ್ಚಿನ ಸಂಖ್ಯೆಯಲ್ಲಿ ಹಾಜರಾಗಿ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮವನ್ನು ಕೇಳಬೇಕೆಂದು ಮತ್ತೊಮ್ಮೆ ನಮ್ಮ ಕರ್ನಾಟಕ ವಿಜ್ಞಾನ ಮತ್ತು ತಂತ್ರಜ್ಞಾನ ಅಕಾಡೆಮಿ ವತಿಯಿಂದ ತಿಳಿಸುತ್ತಿದ್ದೇನೆ. ಥ್ಯಾಂಕ್ ಯು, ವರುಣ. ಥ್ಯಾಂಕ್ ಯು, ಮ್ಯಾಮ್.
Thank you, sir. Thank you so much. Thank you.